Welcome to NEET Biology MCQs educational channel. Are you a NEET aspirant? Get ready to challenge your brain with high-quality MCQs based on the NCERT book. Sharpen your concepts, enhance application skills, and master assertion reasoning and statement-based questions. This is your ultimate MCQ power zone for NEET success.
What is the ploidy of cells in a microspore tetrad formed during microsporogenesis? A. Diploid. B. Haploid. C. Triploid. D. Tetraploid. Correct answer is B. Haploid. Explanation. The microspore tetrad forms after a diploid pollen mother cell undergoes meiosis, which includes two successive divisions, meiosis 1 and 2. This process reduces the chromosome number by half, resulting in 4 haploid microspores. These microspores represent the gametophytic generation. Diploid, A, refers to the original pollen mother cell before meiosis. Triploid, C, and tetraploid, D, ploidies are abnormal in this context and do not occur during normal microsporogenesis. Hence, the correct answer is B, haploid. Which wall layer of the microsporangium directly nourishes developing pollen grains? A. Epidermis. B. Endothecium. C. Tapetum. D. Middle layer. Correct answer is C. Tapetum. Explanation. Among the 4 layers of the microsporangium, the tapetum is the innermost and plays a vital nutritive role. It is rich in cytoplasm and often binucleate, contributing essential enzymes, proteins, and nutrients necessary for pollen development. The epidermis, A, provides only protection. The endothecium, B, helps in anther dehiscence but does not nourish pollen. The middle layer, D, degenerates early and has no direct nutritive role. Therefore, the tapetum is the only layer that directly supports developing pollen, making C, tapetum the correct answer. Which tool would best help differentiate the shapes of anthers from different species? A. Compound microscope. B. Dissecting microscope. C. Electron microscope. D. Phase contrast microscope. Correct answer is B. Dissecting microscope. Explanation. A dissecting microscope, also called a stereomicroscope, is ideal for low magnification observations of surface features, like the size, shape, and attachment of anthers. It provides a 3D view, which helps compare structural differences in intact floral parts. A compound microscope, A, is for higher magnification of thin sections, not suitable for whole anthers. Electron microscopes, C, give ultrastructural details, which are excessive for basic morphological comparison. Phase contrast microscopes, D, are used to enhance contrast in transparent specimens, but not suited for gross morphological comparisons. Thus, B, dissecting microscope is the correct tool. What kind of specimen preparation is suggested for comparing stamens from different species? A. Fixation in formalin. B. Mounting in glycerin on a slide. C. Dry mounting on a Petri dish. D. Staining with iodine solution. Correct answer is B. Mounting in glycerin on a slide. Explanation. Mounting stamens in glycerin preserves moisture and allows flexibility and clarity for microscopic observation without causing distortion or desiccation. It keeps the specimen close to its natural state. Fixation in formalin, A, is suitable for long-term storage but alters tissue texture and is unnecessary for immediate observation. Dry mounting, C, can dehydrate the specimen and is not appropriate for fine structural viewing. Staining with iodine, D, is used for detecting starch, not for observing overall stamen morphology. Therefore, B, mounting in glycerin on a slide is the best choice. In which stage are pollen grains shed in more than 60% of angiosperms? A. Unicellular. B. 3-celled. C. 2-celled. D. 4-celled. Correct answer is C. 2-celled. Explanation. In over 60% of angiosperms, pollen grains are shed at the 2-celled stage, which includes a vegetative, tube, cell and a generative cell. This is the common mature stage ready for pollination. Unicellular pollen, A, represents an earlier phase with no cell differentiation. 3-celled pollen, B, although seen in some species, is less common and includes the vegetative cell and two sperm cells, indicating a more advanced stage. A 4-celled stage, D, does not exist in pollen development. Therefore, C, 2-celled is the correct answer. Which component of the pollen grain wall is resistant to high temperature and strong chemicals? A. Cellulose. B. Pectin. C. Sporopollenin. D. Lignin. Correct answer is C. Sporopollenin. Explanation. Sporopollenin, found in the outer layer, exine, of pollen walls, is one of the most chemically inert biological substances. It protects pollen grains from environmental damage and aids in their fossilization. Cellulose, A, and pectin, B, are structural components of general plant cell walls but are not highly resistant to degradation. Lignin, D, though tough, is found in vascular tissues, not in the pollen exine. Thus, C, sporopollenin is the only correct choice. Why are pollen grains preserved well as fossils? A. Due to cellulose-rich intine. B. Because of nutrient-rich cytoplasm. C. Because sporopollenin resists decay. D. Due to their large size. Correct answer is C, because sporopollenin resists decay. Explanation. Pollen grains are often found in fossil records because their exine is made of sporopollenin, which resists microbial attack, temperature extremes, and strong acids or alkalis. The intine, A, though rich in cellulose, is not as durable. The cytoplasm, B, decays quickly and does not aid preservation. Size, D, has no role in fossilization. In fact, larger pollen may decay faster. Therefore, C, because sporopollenin resists decay is the correct explanation for fossil longevity. How can tapetal cells become binucleate? A. They undergo endomitosis. B. They fuse with neighboring cells. C. Through delayed cytokinesis white heavy checkmark. D. By absorbing nuclei from pollen grains. Correct answer is C, through delayed cytokinesis. Explanation. Tapetal cells often become binucleate due to delayed cytokinesis, where nuclear division occurs without immediate cytoplasmic division. This adaptation enhances metabolic activity needed for nourishing pollen. Endomitosis, A, refers to DNA replication without nuclear division and doesn't lead to binucleate condition. Fusion with neighboring cells, B, is not a normal feature of tapetum. Absorbing nuclei from pollen grains, D, is biologically implausible. Hence, C, through delayed cytokinesis is the valid answer. Why might it be useful to store pollen in liquid nitrogen for crop breeding? A. To maintain their size and shape. B. To improve pollen allergy treatment. C. To enable long-term viability for fertilization. D. To enhance pollen color for identification. Correct answer is C. To enable long-term viability for fertilization. Explanation. Pollen stored in liquid nitrogen at -196 degrees Celsius retains viability for years, allowing breeders to cross plants across seasons or locations. This aids in hybridization and conservation of germplasm. Size and shape, A, are not as critical as viability. Pollen allergy treatment, B, is unrelated to cryopreservation. Enhancing color, D, is unnecessary in breeding and doesn't require nitrogen storage. Therefore, C, to enable long-term viability for fertilization is the most accurate application. Which factor primarily affects how long pollen grains remain viable after release? A. Type of flower. B. Size of pollen. C. Temperature and humidity. D. Presence of nectar. Correct answer is C. Temperature and humidity. Explanation. Environmental conditions, particularly temperature and humidity, have the greatest influence on pollen viability. High heat and low humidity can cause desiccation, shortening the lifespan of viable pollen. The type of flower, A, may influence pollen production, but not directly its viability duration. Size, B, is not a consistent determinant of viability. Nectar, D, has no relation to pollen viability post-release. Thus, C, temperature and humidity is the correct answer. What structure in a flower produces microspores through meiosis? A. Anther wall. B. Tapetum. C. Sporogenous tissue. D. Filament. Correct answer is C. Sporogenous tissue. Explanation. The sporogenous tissue, located inside the microsporangium of the anther, consists of diploid cells that function as pollen mother cells. These undergo meiosis to produce haploid microspores. The anther wall, A, and tapetum, B, are supporting layers. The wall provides structure while the tapetum nourishes developing pollen. The filament, D, merely supports the anther. It is not involved in microspore production. What is the shape of a typical microsporangium in transverse section? A. Square. B. Circular. C. Oval. D. Triangular. Correct answer is B. Circular. Explanation. In transverse section, a mature microsporangium appears almost perfectly circular due to its tetrasporangiate organization and rounded structure. Option A, square, C, oval, and D, triangular, are incorrect because they do not represent the anatomical symmetry observed in a typical microsporangium. Which layer helps in the dehiscence of the anther? A. Epidermis. B. Endothecium. C. Tapetum. D. Pollen sac. Correct answer is B. Endothecium. Explanation. The endothecium, located beneath the epidermis of the anther wall, becomes hygroscopic and helps the anther dehisce, split open, to release pollen. The epidermis, A, provides protection but doesn't aid in dehiscence. The tapetum, C, supports pollen development nutritionally, and the pollen sac, D, is the chamber where microspores develop but does not contribute actively to anther opening. How many JECA are present in a typical angiosperm anther? A. 1, B. 2, C. 3, D. 4. Correct answer is B. 2. Explanation. A typical angiosperm anther is dithecous, meaning it has two lobes or thecae, each containing two microsporangia. Option A. 1. Describes monothecous anthers seen in some monocots. Options C and D are incorrect, as 3 or 4 thecae are not typical of standard angiosperm anther anatomy. What connects the filament of a stamen to the flower? A. Anther. B. Ovary. C. Thalamus. D. Petiole. Correct answer is C. Thalamus. Explanation. The filament, which supports the anther, is attached at its base to the thalamus or receptacle of the flower. The anther, A, is the terminal structure on the filament, not the base. The ovary, B, is part of the female reproductive organ, carpel, and the petiole, D, is the stalk of a leaf, not related to floral structures. What is the name of the process through which pollen mother cells form microspores? A. Microsporogenesis. B. Megasporogenesis. C. Pollination. D. Fertilization. Correct answer is A. Microsporogenesis. Explanation. Microsporogenesis is the process in which diploid pollen mother cells, microsporocytes, undergo meiosis to produce haploid microspores. Megasporogenesis, B, refers to the same process in ovules, not anthers. Pollination, C, is the transfer of pollen, and fertilization, D, is the fusion of male and female gametes. What is the function of germ pores in pollen grains? A. Nutrient absorption. B. Gas exchange. C. Site of pollen tube emergence. D. Waste removal. Correct answer is C. Site of pollen tube emergence. Explanation. Germ pores are thinner areas in the tough exine layer of pollen grains where the pollen tube emerges during germination. They are not involved in nutrient absorption, A, gas exchange, B, or waste removal, D, which are cellular or tissue-level functions unrelated to the structure of pollen. What is the usual diameter range of pollen grains? A. 10 to 20 micrometer. B. 20 to 30 micrometer. C. 25 to 50 micrometer. D. 50 to 100 micrometer. Correct answer is C. 25 to 50 micrometer. Explanation. Most pollen grains fall within the 25 to 50 micrometer range in diameter. Option A underestimates the size, while options B and D either partially overlap or overshoot the average range seen in flowering plants. To observe cytoplasmic details in pollen grains, which stain would be most effective? A. Safranin. B. Methylene blue. C. Acetocarmine. D. Iodine solution. Correct answer is C. Acetocarmine. Explanation. Acetocarmine stains chromatin material and cytoplasm, making internal structures of pollen grains clearly visible. Safranin, A, primarily stains lignified tissues. Methylene blue, B, is better for general staining of cell nuclei, but less effective here. Iodine, D, reacts with starch, not useful for pollen cytoplasmic observation. Which method helps identify binucleate tapetal cells? A, longitudinal section of filament. B, TS of microsporangium. C, viewing exine under microscope. D, examining pollen viability. Correct answer is B, TS of microsporangium. Explanation: binucleate tapetal cells are observable in transverse sections of microsporangia under a microscope. The filament, A, does not contain tapetal cells. Observing the exine, C, shows surface patterns, not cellular nuclei. Viability tests, D, assess pollen health, not tapetal structure. To observe pollen tube emergence, which setup is most suitable? A, dry pollen on Petri dish. B, pollen in distilled water. C, pollen on germination medium. D, pollen in alcohol. Correct answer is C, pollen on germination medium. Explanation: a germination medium containing nutrients like sucrose supports pollen tube emergence. Dry pollen, A, won't germinate. Distilled water, B, lacks nutrients needed for tube growth. Alcohol, D, kills cells, so pollen can't germinate. Which technique best visualizes sporopollenin on pollen surface? A, light microscopy. B, phase contrast microscopy. C, electron microscopy. D, X-ray crystallography. Correct answer is C, electron microscopy. Explanation: electron microscopy provides the resolution needed to see sporopollenin in the exine layer, which forms the tough outer wall of pollen grains. Light and phase contrast microscopy lacks sufficient detail. X-ray crystallography, D, is not suitable for irregular biological surfaces. Which part of the flower should be sectioned to study microsporogenesis? A, stigma. B, anther. C, filament. D, style. Correct answer is B, anther. Explanation: microsporogenesis occurs in the microsporangia of the anther, so it must be sectioned for microscopic study. The stigma, A, style, D, and filament, C, are unrelated to microspore development. How would you determine if pollen is viable after release? A, microscopic shape. B, number of pores. C, staining and germination tests. D, size of grain. Correct answer is C, staining and germination tests. Explanation: pollen viability is confirmed through staining, example acetocarmine or fluorescein diacetate, or germination tests on nutrient media. Microscopic shape, A, pore number, B, or size, D, don't confirm viability. Non-viable pollen may still have normal morphology. Which microscopy helps observe pollen wall design? A, bright field microscopy. B, polarizing microscopy. C, dissecting microscopy. D, scanning electron microscopy. Correct answer is D, scanning electron microscopy. Explanation: SEM provides high-resolution 3D images of the pollen wall's sculptured surface. Bright field, A, polarizing. B, and dissecting microscopy, C. Lack the resolution to reveal detailed exine patterns. Which feature of sporopollenin contributes most significantly to the fossilization of pollen grains? A. It is acidic in nature. B. It is resistant to enzymes, acids, and alkali. C. It contains cellulose. D. It is hydrophilic. Correct answer is B. It is resistant to enzymes, acids, and alkali. Explanation. Sporopollenin is a key component of the pollen exine, making it highly durable and resistant to degradation by extreme chemical and biological agents. This feature allows pollen to survive in the fossil record for millions of years. The NCERT clearly states that no enzyme has yet been found that can degrade sporopollenin. Option A is wrong as sporopollenin's resistance isn't due to acidity. Option C is false since exine does not contain cellulose. Option D is incorrect because sporopollenin is not hydrophilic. Which of the following is a correct sequence of development in the anther? A. Microsporangium, pollen sac, sporogenous tissue, microspores. B. Sporogenous tissue, microsporangium, pollen sac, microspores. C. Microsporangium, sporogenous tissue, pollen sac, microspores. D. Sporogenous tissue, pollen sac, microsporangium, microspores. Correct answer is A. Microsporangium, pollen sac, sporogenous tissue, microspores. Explanation. The correct developmental sequence in the anther begins with the formation of microsporangia, which then become pollen sacs. These sacs house the sporogenous tissue that undergoes meiosis to produce microspores. This is the exact flow described in the NCERT. Option B and D place the stages out of order, while C places the sporogenous tissue too early in the sequence. Thus, A is the most accurate and logically ordered option. Why are pollen grains of Parthenium a serious health concern in India? A. They are genetically modified and allergenic. B. They trigger autoimmune disorders. C. They cause severe respiratory allergies like asthma and bronchitis. D. They contain toxic alkaloids absorbed through the skin. Correct answer is C. They cause severe respiratory allergies like asthma and bronchitis. Explanation. The pollen grains of plants like Parthenium, carrot grass, cause severe allergies, often triggering respiratory conditions like asthma and bronchitis. These responses are due to hypersensitivity to airborne pollen proteins. Option A is incorrect because there is no indication of genetic modification. Option B is false. The problem is allergy, not autoimmune disease. Option D is incorrect because the issue is inhalation, not skin contact. The generative cell divides within the pollen grain in some species. Which process follows this mitotic division? A. Formation of sporogenous tissue. B. Development of two male gametes. C. Dehydration of microspores. D. Pollen grain in time thickening. Correct answer is B. Development of two male gametes. Explanation. In species that shed pollen in the 3-cell stage, the generative cell divides mitotically within the pollen grain to form two male gametes. This is a key step in the angiosperm reproductive process. Option A is unrelated to the pollen grain stage. Option C refers to microspore maturation, not gamete formation. Option D is wrong because intine thickening is not relevant to the generative cell division. Which feature of the pollen grain wall ensures its survival in extreme environmental conditions? A. Pectin in the intine. B. Cellulose in the exine. C. Sporopollenin in the exine. D. Multinucleate cytoplasm. Correct answer is C. Sporopollenin in the exine. The durability of pollen grains is primarily due to the presence of sporopollenin in the exine. It resists degradation by high temperatures, acids, and enzymes, making pollen resilient in extreme environments. Option A, pectin, and B, cellulose, refer to intine, not exine. Option D is incorrect since multinucleation of the cytoplasm doesn't contribute to durability.